Hi, welcome to the BYU-Idaho Tutoring Center videos. The topic of today's video is passive transport. Now, two really important things that distinguish passive transport from active transport, which we talked about in another video, is that passive transport does not require energy, and passive transport moves things from high concentrations to low concentrations, or down their concentration gradient, which we're going to talk about in a second. So actually, let's talk about right now. So concentration gradients simply describe the region along which the density of a chemical substance decreases. So what does that mean? It just means that you have some sort of barrier, or it doesn't even really have to be a barrier, but some sort of dividing line in which there is more of some molecule or substance than on the other. And naturally what happens is the side that has more wants to diffuse and spread out evenly. And that's where the word diffusion comes into play. It's just spreading things out evenly. So for example, if you've ever sprayed some sort of perfume or Lysol spray in the air, you'll notice that if you're standing on one side of the room, at first you can't smell it, but over time it's the smell or the particles start to diffuse to where you are. So that's basically diffusion. Osmosis is just a diffusion of water across a membrane. And so it's just more specific to water. And I talk more about osmosis and tonicity in another video. So if you need more help on that, go watch that video. So there's two types of, passive transport. There's passive diffusion and facilitated diffusion. So passive diffusion is just molecules or substances diffusing right across a biological membrane. And so this only works for small hydrophobic molecules. Because if you remember, the cellular membrane is made up of phospholipids, which have these hydrophobic tails and hydrophilic heads. And so if anything wants to be able to pass through here, it has to be okay being in this hydrophobic environment for a little bit. So, and it also has to be small because if it's too big, it's not going to be able to make it through. So some examples are CO2 and oxygen. Those are some of the big examples. Water actually doesn't go across the membrane by passive diffusion. It actually goes across by what we call facilitated diffusion. And so facilitated diffusion still isn't going to use energy and still is going to transport molecules from high to low. However, it's going to use membrane proteins to do so. And there's two types of membrane proteins, channel and carrier. And so the way that water diffuses across the membrane is actually by a channel protein, which we call an aquaporin. So make sure you know what that is. So here's a picture of a channel and a carrier protein. So channel proteins, they just let the solutes go from one side to the other. So if you notice, we have a higher concentration of our blue molecule down here than up here. So the blue molecule's just going to go through the channel protein up to the lower area until we have about equal concentrations of each on each side. Carrier proteins, on the other hand, what they do is they take in a solute and then they have a conformational change or shift, and then they release the solute on the other side. So what that would kind of look like is this molecule right here would have a sort of shift, if you would. Sorry about that. And it would release the solute to the other side. So it kind of just changes positions. And then because this is a low concentration, it probably won't transport anything back up here. So we change positions again, take another one down. And so this is slower because it actually has to switch back and forth between the two different conformations. So that's it for facilitated diffusion. Real fast, I want to talk more about concentration gradients. So we talked about how it's difference of concentration of solutes, but it can get more specific and we can talk about electrochemical gradients. And electrochemical gradients take in consideration both the concentration gradient and the membrane potential. So membrane potential has to do with the difference of positive or negatives on either side of a membrane. So in this example down here, we have these red solutes that are positively charged and these blue solutes that are negatively charged. So when we tally everything up, we have 1 blue on this side and 3 red, so that's talking about the concentration gradient. And we have 5 blue and 4 red, and so that's also talking about the concentration gradient. And then it has an overall charge of +2 on this side and -1 on this side. And so if you look at the red, you would think, there's more red on this side than this side, so maybe the red wants to come over here based on concentration gradients. But if you notice, it's actually more positive on this side. So if we're transporting more red on this side, we're also transporting more positive. And so you kind of have this disagreement type thing going on. And so then ATP might actually be required to pump more red onto this side because we've been making it more positive. And we'll see that in a little bit with active transport. Active transport is using ATP or energy to pump things from low to high. And it makes sense that it doesn't require energy. I always like to use this analogy. So if you have somebody on top of a hill, they're up high, and if they drop a ball, the ball will roll down the hill naturally. It doesn't require any input of energy, so it's going from high to low. Whereas if you wanted to get the ball from the low to the high, you are going to have to use energy. And that's again where active transport comes into play. That's all for today's video. I hope you better understand the difference between passive diffusion and facilitated diffusion. And in another video, I'll go into active transport and the different components of that.